right, hello everybody, and welcome to another AgroPlus webinar. Today we have part two of the Taurus webinar with Clayton Miller and Mike Dolinsky. Yep, almost lost
Yep,
your last name there,
that's okay.
but yeah, we went a little long last time. Clayton is obviously very passionate about what he does, so we're gonna give Mike his own webinar here to talk about his foliar abilities here. So we're gonna let him get to it. Take it away, Mike.
You bet. Well, welcome. And this is going to take us close to that hour that we spent last week here to get through this. So sit back and you're going to be able to get exposure to this when anytime you want here. I'm going to be posting a big chunk of this on Taurus's website. So you'll be able to look at it. And I'm a believer that unless you understand how a plant grows, and most people are not being educated on that anymore. There's no extension people left in the country, and retailers in many cases are concerned with selling you products. So we want to provide this for our clients. And I'll just get it going through the story as I tell it, and then we'll wind up on foliars and so on. And I hope you'll see the connections. Here in the West, we put the seed in the ground. By the way, can you see my cursor when I'm running it here? Yeah, okay, because I point to things a lot. Uh, we shove the seed in the ground. We let nature look after it for the summer. We harvest it. Then we shove it into the bin, and then we ship it somewhere. And we ship all those nutrients away, micros, macros, and so on. And we do that in about 90 to 120 days. So it's a tough life we have here. Now, supplying nutrients at key stages is critical. And I'm a firm believer in this. And if you've been at any of my webinars over the years, uh, that we have to understand the cell. If we look after photosynthesis and we look after energy production and stress management, as we saw in my title, the plants know what to do. So we're going to look through this whole thing, because if you understand this, I think you'll be able to make much better decisions about what you're going to do with your nutrients. Now, I use this a lot, and you guys probably in flatland country down south there in southern Alberta never saw a hill this high until you got to the mountains. But I use this. This is northern Saskatchewan, and it tells a story. And for you guys, this gentleman right here is Phil Thomas, and he passed away at the beginning of 2020 and during the COVID period. And Mr. Canola. Now, I use this because it tells an important story that maybe farmers don't think about. But you would probably agree that the plants up here at the hilltop are in a different environment, both in the air and in the soil, than the plants down here or the plants right here. The reality is plants matter of inches apart from each other in different environments, soil-wise, because of organic matter, pHs, nutrient distribution, and so on. The other key component of understanding this is that all these plants are genetically the same. So that asks, you've got to ask the question, well, how is it that each one of these plants growing in this different environment can sustain themselves? And the fact of the matter is it's through genetics. By being able to produce proteins, produce things that the plant needs in order to deal with all of those environmental conditions. And that's what we'll be talking about. What are those environmental conditions and how do they do that? Well, let's take a look at genetics, because if it's all about genetics, we better have a look at something here. Here's the human genome, somewhere between 24 and 25,000 genes. Now, proteins that are produced by genes primarily can be modified after. So they're not finished that. But look at wheat. In the whole wheat genome, there's about 108,000 genes. Napis, that's canola as we know it, about 101,000. And then you look at the rest of them, 44,000 in peas, 45, barley, 30, what do we got? Potatoes, 29, chickpeas, 28. First message is, reason we grow wheat and canola, it's got horrendous genomes. It can do lots of things. The human body is simple compared to what goes on in those plants. And the reason is that their history, their epigenetic history and their evolutionary history. Now, when I do these kind of talks, I also like to do basic biology because, I want to educate people generally. This is, base pairs, this is DNA. And DNA is made up of a sugar right here, it's five-sided carbon, a phosphorus, and then these are nitrogen. Now, the first message I leave you is that the first thing a plant has to do is build a cell. And to build that cell, it needs nitrogen and it needs phosphorus because the phosphorus goes into all the DNA and so does the nitrogen. Nitrogen is the largest amount of nutrient in virtually all plants because that's what's used to build the cells. So your plants never want to be out of nitrogen and they don't want to be out of phosphorus because the phosphorus provides the energy. So I put this in here, and unfortunately, let's see if I can get rid of this picture here because there, what happened? Can you see it? I can't see it anymore. Oh, man. Where did you go?
You're still here, Mike.
Yep.
Okay, I guess I got to leave it there. Mitochondria or ATP, adenosine triphosphate. People, we always talk about ATP, but we never explain what it is. That's really the source of energy in all life on this planet. And it's the ATP that drives everything in terms of energy in the plant, and it's key for that. So we got to take a look at, what's going on in the plant's life cycle, and the energy is related to a couple of things which I'll cover in a minute or two. But we really got to understand, what does it take to grow a plant? Well, it takes CO2, comes from the air, and we need that because it's the only way we get carbon. And if you think about it for a minute, we as human beings or animals or cattle, whatever, we eat plants or we eat the animals that eat plants to get the carbon. And the only thing they make from this carbon is sugar, glucose, sucrose, and so on. And then that's used for energy, just like in the human body, we use glucose. But the plant doesn't eat anything. It makes it all from this. All we do and animals do is we eat the plants or we eat the animals that eat plants and we get that carbon. Water is important for a lot of reasons, and water drives everything because plants drink, they don't eat. So it's got to be soluble. Oxygen, well, we can't change oxygen much. You can do something with water. You guys in the South have to deal with water problems all the time. So you irrigate, but on dry land, you can maybe adjust a little bit. You can have your stubble straw, a little higher. You can conserve moisture. One of the biggest breakthroughs for prairie agriculture was going to minimum till. What that did is conserve moisture, which gave you guys in the South dryland country a big advantage. I came to Alberta in 1972. Canola wasn't grown south of Highway number 1. We were growing Polish canola because it was too dry and too hot. You had a lot of years where there was good moisture. You're growing 14, 50 bushels of canola on dry land, which was thought impossible in the early 70s. Yields were 16 bushels an acre as a provincial average. Now we're struggling at 35. 50 years later, we're sort of doubled our yields and we want to go to 50. So oxygen, we can't do much about. Sunlight, well, we can't do much about it. In fact, when we get those smoky days, maybe we haven't got enough. Microbes are important and we won't cover that. But really, what do you have control of? Nutrients. So my message to you, a second message, you better really be good at using nutrients and managing nutrients because that's all you got control of on the whole. So back to that, photosynthesis, that produces the sugar. The only thing that produces the sugar is photosynthesis. Stress, we'll talk about at the end. We build the cells, and I'll show you that. We got to feed every plant, and that's what you're trying to do. Remember, you got variability across that field, real variability. So the only way you can feed them is to put nutrients generally across the field in positions. You put a lot of them here in the seeding, the germination here at the beginning. You put a lot of those macros in there because it takes a large volume to build the plant. So you got to put a fair amount in there. During this growing season, as the plant takes off through its rapid expansion, all you can really do, you can put nitrogen down anytime, but you can't really put down a lot of granular phosphorus because it has no way to move in. You can't put potassium as a granule in because you don't get enough rainfall and you can't leach them into the soil. So you're stuck with putting on foliar treatments to help that plant through these rapid expansion times into reproduction. And reproduction is key. We got to build this nutrient in the plant before we hit reproduction. Because what happens, it converts from this vegetative production, and vegetative production is not about leaves. It's growing all its reproductive parts through this whole process. And if you short plants of both nutrients and water and sunlight and everything else, by the time you get reproduction, you're hooped. You can't get much more into the plant because hormonally the plant changes. And I'll cover this again. This is sort of a synopsis in a nutshell. But during this growth phase here, the plant is producing sugar and feeding the roots and the leaves and building it. When it hits reproduction, what happens? The focus or the sink now becomes the seed. So the plant cuts back on the amount of nutrient it's or photosynthesis assimilates, is feeding the roots. And here's the message: the roots can't feed themselves. And that's really important because they have no way to produce sugar. They can't photosynthesize. So it's the shoot that supplies the nutrients to the root. And then we go through grain filling. And with wheat, you should know that by the time you hit reproduction, the filling of the seed, 70% of the nutrients and material going into filling comes from the plant shoot and roots that it's extracting itself. It's not taking much more in at that point in time. And we have to have the water that follows that. We get this build, and then we fill, and the water follows that. Now, I threw this in right now because, we do sell products, and you'll take a look at when we put on nutrients. Here's active prying. We put that on the seed. Okay, look what's in there. NPK. Look at the phosphorus in there. That's on the seed. Boron, iron, manganese, zinc. When we go into the growth phase at herbicide time, we put on more phosphorus, phosphate, NPK. We put on manganese, molybdenum, zinc. And I'm just pointing these out because these are the things I'm going to be talking about as we move forward. Then we get into fungicide timing or filling. We put on N, no more P. We don't need a whole lot more P because the plant is largely finished growing, building cells, and now it's into filling, going to be filling. And K, because K is involved in movement of everything in the plant. You never want to run out of K either, to tell you the honest truth, and I'll get into that. Sulfur and iron. So these are the things we put on, and that's just in wheat. And we have different products for different crops because different crops need different nutrients. Now you're struggling with all of these things: stratification, cold soil, salt index problems, pHs, compaction, you're looking at tissue test, you've got zone management, you're doing all kinds of things to try and help your crop. And you should. And when you see the webinar that will eventually be posted, there's a lot more factors in there, but you're doing your best. And I'm really concerned about stratification and so on. But let's take a look at variability. This is a map of phosphorus levels across the prairies. Red is bad. You can see that. So we know we got variability. This is zinc. That's about the same thing. Well, why is that happening? Well, where the heck have we been farming for a long time? 100 years? Manitoba more so, but they got pigs and more cattle and heavier land. This is the brown soil zone, largely. We've pulled out piles. Look at the stratification that's happening in your fields. Organic matter, this is inches. As we go down in the soil profile, you can see organic matter reduces. Look at your phosphorus levels. By the time you get down 2 to 4 inches, your plants are struggling to get phosphorus. Why is that? because they pulled the phosphorus out of there and we can't get it back down there. All that phosphorus is in the top 1 to 2 inches because it's immobile in the soil. You can't leach it down, and you guys in the south don't get enough rain to hardly grow a crop in many years. So how are you going to leach down nutrients below that surface? And we keep putting more phosphorus right there with the seed all the time. Potassium's the same thing. It's immobile largely in the soil, hardly moves. Again, no rain. Zinc. Well, zinc's a metal. So once you pull the zinc out, unless you put some back, eventually you're going to run out. And you saw that map of zinc deficiency. Manganese is the same thing. It's a metal, transition metal, really important. This is out of Claire's home. This Ross McKenzie did this. Look at the minimum and maximums. Just taking samples, going across a field. Look at the phosphorus levels. I was saying, differences between inches between plants and the levels of them. Potassium levels, horrendous differences. Sulfate levels, pHs. Look at this, 2-point pH difference in the same field. Yes, Clayton.
So then on that last slide, when you had the big accumulation of phosphorus in that 1 inch zone from the top, then if guys are seeding 1.5 inches deep all the time, right? We're not usually hitting that 2 inch too often, right? If we don't have to. So how long does it take to leach 1 inch in the soil profile then? It all depends on moisture, right? But to get it down to that, like we always sample phosphorus in the 0 to 6 inch zone. And that's mainly why, because it's not a mobile nutrient. But how long does it take to get down that deep if you're only seeding 1 inch all the time?
Probably never.
Probably never.
Probably never. The thing is with phosphorus, it's a cation, surprisingly, which tells you that it should move, but it doesn't move because it gets tied up so darn fast by calcium, magnesium, manganese, and iron. So once it's tied up, microbes have to break it off, and the root enzymes have to break it off. And I'll show you this in a minute. Just hang on a sec. But take a look at this pH, which is really important. A 6 to almost 8, 2-point difference running across that same field. And organic matter, which you won't worry about. But here's your answer, Clayton. This is soil sampling here at Camrose. Take a look at, where's Foss here? 42, 22, 9 in the 6 to 12 inch zone. Look at potassium. Because we take the crop up, we take the trash, we put it back on top, and it's dry. So we send off some with the seed. The rest of that, the plant has, all that's left in the ground is the root and the exudates. That's all that's left there. And the plant extracts most everything from the root as it's going into maturity. When you take a look at a root on a wheat plant or a canola plant, it's sucked dry by the time it's harvested. Look at zinc, doesn't move as we go down. Why does the plant send roots down beyond 10 inches? I'll show you more stuff. They're good questions. Only one reason it goes down deep. Because what do you get when you go down deep? You got no oxygen.
Moisture.
You got no minerals. You got water. It's the only reason it goes down deep. Because it costs. Remember, whenever the root has to go down, the shoot has to feed it. So if your plant is spending all of its time growing roots just to survive, to get water, you're not going to get a crop worth a damn. That's why drought is so bad, because the water solubilizes nutrients. The plant has to have water in order to transpire and send it around, but I'm getting ahead of myself. Let's take a look at the cell. So here's the trick. And this is where I'm going. We have light that affects the chloroplast, which makes sugar, which is then fed. And this is the key point that I've been missing for years. It's fed to the mitochondria. And it's the mitochondria in the cell, and I'll explain this, so don't panic about mitochondria, because, I'll get to it. It produces ATP, and then the CO2 gives off, and it goes. And that's all driven by light. And the real neat thing is plants are what are called autotrophs. They're the only organisms that can make everything from just what they get from the atmosphere and the soil. All life on the earth depends on autotrophs. The bacteria, fungi, everything that are called eukaryotes depend on that. So we have to make sure we handle photosynthesis, because without that, nothing much is going to happen good for you. It's just that simple. And here's a cell. You can see you're going to be going back to university on this thing, guys, whether you like it or not. And if you never went to university, some of this may seem scary to you. And you don't have to remember this effectively. You will never see any of it unless you look inside a cell. But this is what's going on. And if you understand this, you'll know why I say the plant knows what to do, because this is all run by genetics. And the plant has sensors that is sensing everything. So here we have the mitochondria. Here we have the chloroplast where photosynthesis takes place. Here's the nucleus where all the genetic material hangs out. And then there's a bunch of other stuff. But look at this. And there's a vacuole. And a vacuole is important. It's like a fat cell in a human being. When the plant has too much nutrient, it stores it in the vacuole, and then it expands. It can move all these other organelles around because they're just floating around inside the cell. And then when it runs out, it'll give it back to the liquid part, the cytoplasm, and continue growing that cell. So when there's water, it stores it up there. When there's excess nutrients, say when a root hits a sideband or a mid-row band, there's an excess of nutrients. It doesn't have to spend much energy to get those nutrients from there compared to searching through the soil, looking for a granule here and a particle there and so on. That's why side banding, mid-row banding, and concentration of nutrients is really efficient, because the plant roots then don't have to spend a lot of ATP in searching and extracting those nutrients. But look at this. All these nutrients, zinc, copper, magnesium, iron, more copper, manganese, molybdenum, zinc, they're in every one of these organelles. And the plant's job is to make that plant stable, homeostasis, balance everything. And it does it because it's got a cell wall here. I'll use this photograph here. This is the cell wall. Here's the nucleus. Here's the chloroplast. And I can't see the mitochondria because the picture's in here. Wait a minute. If I get this, will this? Oh, I got it. Here's the mitochondria. So the nucleus has all the messages, and it sends out what are called microRNA and messenger RNA, and it makes proteins. Now, here's the thing you want to take a look at. See this passageway here? These are called plasmodesmata. It's not, that's just really cool. And they only discovered these in about 1995. So we now know that the plant can send messages between the cells. They're called microRNA. They can send messages and form a response to, for example, a disease or something like that. So this is what's going on in the cell. We need all these nutrients all the time in the cell. So here's the cell and wall, and these are, all this is phosphorus by a fat here, lipid layer, and then phosphorus. And then the plant puts these little sensors everywhere there, and it controls everything going into the cell. You can see here the cations and anions. One thing I'll point out for you guys, urea. Urea is uncharged molecule. Urea goes in through the cell wall like a dart, and so does water. These nutrients are controlled. They're controlled by these transporters in the cell wall. The plant controls everything going in and out through these transporters. Even getting it from the soil, they'll put out exudates into the soil to kick off things like phosphorus and potassium, and they'll extract them this way. Even the water is run by proteins that control water. And you can see that when you see this little structure here with the blue dots and these lipids, that's the plasma membrane, okay? And they put these little transporters, here's nitrate, ammonium, sulfate, in the roots. Each cell everywhere can do this. And that's how they extract those nutrients. And then here's photosynthesis. They take those nutrients, run them up the plant into the leaves, and the sun makes sugar right here, breaks water down, gives off oxygen, luckily for us, with water going in, produces a little bit of ATP here, and then it goes to the second part of photosynthesis here called the Kelvin-Benson cycle. And here's where it takes in CO2, runs it on this enzyme called rubisco with some water, and lo and behold, you get sugar. That's all photosynthesis does. And this little bit of ATP produced there is only used in the chloroplast. It's not used to energize a cell. But I got to show you something important. And you know, this is really cool because life on Earth depends on this. Water, the first thing that the plant has to do to have photosynthesis is break the water molecule right here. Oxygen becomes a waste product and it needs the hydrogen to drive the production of ATP. It's this, and it's really interesting, and I wish I could explain this to you a little bit better, but it's like a little windmill. The hydrogen here pressurizes it on this side of the cell, and it turns this turbine and it hooks a phosphorus to ADP to produce ATP. It's absolutely amazing. And then this electronic transport chain from light in the photosystems transfers this energy along here to help make that ATP. But I got to show you what's going on here. You got this stuff in the electron transport, and I don't need to explain what happens. It just energizes, it moves along, and accumulates these hydrogens. Plastocyanin, cytochromes, ferredoxin. What are those things? Well, here they are. This cluster is what breaks down water right off the bat. Manganese, calcium, and oxygen. Plastocyanin, copper, sulfur, nitrogen. About 25% of all the copper in the NA plant, all plants do this, photosynthesize, are used for this process. Ferredoxin, iron, sulfur, cysteine, this amino acid, this is methionine is another sulfur amino acids. I point this out so we can understand why just putting on macronutrients without all the micronutrients, because these are all micronutrients, they're metals, they're catalysts, that's what runs the plant. Here's magnesium and nitrogen in chlorophyll. And here's simply how it works. Photosynthesis gives us sugar, goes into respiration, makes ATP, and out of that sugar we make all the amino acids, all the carbohydrates, and all the fats. I hope that by going through this agonizing thing, that you appreciate how important photosynthesis is and the nutrients that are in photosynthesis and the nutrients that are used everywhere in the plant. And here's the way the plant handles it. Takes up sugar and nutrients and water into the leaf, up the stem, into the leaf, runs it through photosynthesis, kicks out the oxygen, makes sugar, and moves it back down to the roots, moves that sugar back up to the apical meristem, and they do that and communicate between the tip of the plant and the root tip using hormones. Auxins, gibberellins, abscisic acid, jasmanic acid. All these hormones are messages that the plant uses to bring about action. Now we get down to some real stuff. This is canola. So what does that apical meristem look like? Here are the cotyledons right here. These are the two cotyledons on canola come up. This is right in the base of it. This is called the growing point. This is the first true leaf. That's the second true leaf. And that growing point will continue at the top of the plant and keep going until you get all the pods develop. Here's inside the clam leaf on peas, if you look inside. Notice we have all these tendrils already formed here. And all the way here, there's like about 5 different tendrils in here, and there's the growing point. So each one of these then will come out on a node, and there goes the pea plant. If you lose this growing point, the plant will die. That's why frost at this stage on canola, if that's black, plant's dead, because everything there's nothing down in the soil. In peas, if it freezes off, because the the cotyledons don't come up. They stay in the ground. It can regerminate. But with canola, all the energy's up. There's nothing left down below. It'll die. And this is wheat, the tip of a wheat plant. In the roots, the plant gives, breathing or respiration. The plant gives up carbonic acid, acidifies the soil. You need to acidify the soil here on the cation exchange, and this is a little complicated, but Lyle will understand this. The cation exchange is negative in the clay particles, and the soil is negatively charged. The cations are positively charged, so they hang on to the cation exchange. If you acidify this and put on positive charges, it'll kick off these other ones. That's called the cation exchange. Those go into the soil solution, and the plant, by pulling water in, will pull a certain number of nutrients to it, but potassium and phosphorus don't move along. So it goes in through the root hair, into the xylem, up the plant, into the leaf. The water comes out the leaf, and that puts suction on the roots, and that cycle goes on. It's like a siphon. It's losing it there. There's water pressure on the below, and it's moving into the roots, and out comes there. The other thing is the CO2 goes in, and the oxygen comes out. And when the stomata closes because of stress, it's dangerous. The oxygen that is trapped becomes toxic to the plant. And water leaving drives that whole process called transpiration. Very simple. What happens, though, when it's dry and hot? Well, the plant's in a bind, because if it keeps using water, it's going to run out. But if it stops using water, it's going to cook. If it stops moving water, it doesn't feed, and it doesn't get nutrients that continue growing. That's why drought is really hard on the plant. And potassium controls the opening and closing on those stomata. Potassium, chlorides, there's a lot of other things, but this simply shows that it's water going into these guard cells and leaving the guard cells that open and close them, and that happens really, really quickly. And those assimilates that are produced, the source, it's called the source, from CO2, minerals, and so on, goes to the sink, which are the root tip, a tuber, a root, a leaf up in the upper part of the plant that are short of nutrients. So there's always a source to sink relationship. The leaves are the source, the sinks are everything else. So how does a plant move it after it gets the sugar developed? Well, let's take a look. We know that the plant has two systems, just like a human being. I think, it's the same, a venous system and an arterial system. The arteries are what used to pump the oxygenated nutrient stuff, and the veins bring it back to the heart and recycle it. Well, the same thing happens in the plant. We're all eukaryotes. We use the same amino acids. We use the same nutrients. We even have same systems almost. So nutrients with water come up the plant in the xylem, but when we have sugar produced, we get the sugar that moves into these companion cells through those plasmodesmata, and then that moves into the phloem. Now, when you put a lot of solutes like sugar into a solution, it's got a high concentration of sugar and a low concentration of water. Water will move from a high concentration, a low concentration, let me get this straight out. It's drawn into here because this is a low concentration of water, and here it's a high concentration of water. That pressurizes the cell, and here's a cell that needs it. So there's a difference in pressure. So it moves down, goes back into these companion cells, moves into the sink cells where it's needed, whether that be a tuber, a root, or the apical meristem. The water then moves back in through portals into the xylem. So you can see that water is key to pressurizing it and moving all these nutrients. And this happens, and you should know this, if you're going to put on any nutrients that you think are going to go in through the stomata. Here we have the stomatal opening. Starts off with the sun. When the blue light from the sun hits the plant, it starts opening its stomata. Okay. The green stuff here is potassium. The potassium starts rising and being pulled up as the stomata opening. And then the stomata are open by about 1 o'clock maximum. The plant is going full tilt. And you can see here that we look at the osmotic pressure in the stomata. At the morning, it's low. By 11, it's really high and rolling, and then it tapers down. And by sundown, it's back to where it sort of stays overnight. And then the sugar starts galloping like this. And later on, because now the stomata are open, the potassium's there, nutrients are flowing, and then it goes downhill. So if you're going to put on a foliar nutrient and you want to get it in through the stomata, you probably want to go sometimes here, 10 o'clock, 11 o'clock, depending on temperatures and so on. Roots and shoots. Let's look at roots. These are some guy, and I found this somewhere, actually figured out how many kilometers of roots there are per meter squared. So look at these barleys, got very small roots. Faba beans, huge plant, small roots. Maize, not bad roots. Potato, puny roots. Rape, look at the roots. Remember those genes that we had? Look at the genes that are associated with roots. Soybeans, small roots. Wheat, big roots. There are good reasons why we grow canola and we grow wheat. They got great roots. Now, how deep do the roots go? This is done at Swift Current, so it's kind of dry land country like you guys. And just in case you're not in this conversion thing, 15 centimeters is equal to 6 inches. So 50% of the roots in most plants are in the top 6 inches. To get 95% of the roots, you go down to these depths. It takes a long way to grow the remaining 95%. And then that remaining 5%, you can see it's got to go down a long, long way. And that's costing the plant a lot of sugar that could be in the shoot. But, what can you do? You generally notice that pea plants don't have very strong roots, all the legumes. And the reason is, I think, is because they always had association with rhizobium and mycorrhizae. So they had partners to help them produce, to extract nutrients, and they didn't have to have those big roots. About 70 to 80% of all the nutrients and water go in through the root hair, and it gets into the plant and goes through these little plasmodesmata from cell to cell to cell. And there are vacuoles in there where they store some all along the way. And eventually they have to go through the cell, through this barrier called the Casparian strip, because the plant has to control what's going into the root. And then once it gets in, then it's off and running. Up the xylem it goes, water and the nutrients. One thing to note is the root hair is a single cell. The only other single cell is the pollen tube in the plant. And the single cell only lives about 5, 6, 7 days at most, depending on conditions. So if the root isn't going through the ground, it doesn't get access to new nutrients that are immobile. That's why phosphorus and so on, they say, if you want to get phosphorus, you got to grow roots. Well, you got to have phosphorus to grow roots to get phosphorus. But once you get the plant healthy and the roots expanding, it accesses a lot of nutrients that a poor, weak root can't, besides the phosphorus. But how does the root grow? Well, this is a weak plant root. And where the stem cells are produced, the meristematic cells are produced right near the tip, but not at the tip. And there's a root cap that covers it. There's a zone here called the elongation zone, which is pressurized by hydraulic pressure. And it's the cells that are born here, so to speak, that are expanded and will drive the root through the ground. That takes place here. The root here is formed behind that in the maturation zone, because if they were down here, they would be ripped off as they move through the ground. After that, the lateral roots come out, and the plant expands its exposure to the soil through the production of all those lateral roots. And here you can see this is the sort of the tip covered by border cells. And here you can see the root. And this is called the rhizosphere, where the plant can extract the nutrients, the immobile nutrients, because the nutrients like phosphorus and potassium don't really get solubilized in water very easily and move to the root. The root has to move to it. And it very quickly will extract the soluble phosphorus from that rhizosphere. And if it doesn't keep growing through the ground, it has trouble getting that phosphorus. And to extract that, it puts out exudates, acids, and so on, which are called phytases and so on for phosphorus purposes, to extract and solubilize that phosphorus out of the organic matter in terms of mineralizing it or kicking it off the cation exchange. It's not a cation, out of the soil itself. So here's how the roots, lateral roots are developed. This is amphaba beans. And here's the vascular system, the xylem and phloem. And you can see the roots are born right here. And the plant is heading towards nutrients here. It actually senses where the nutrients are, so it grows the roots where it wants it. When it gets close to the exterior of the plant opens up the cells, and out comes the lateral root. If you look at the lateral root through cut, you can see that the vascular system goes right back here into the xylem, so that when the plant lateral root picks up nutrients through the root hairs, it immediately feeds back in here and moves it up. So if you have the energy and the sugar to build those plants and those roots, that's when you get a crop. And you need water consistently, unfortunately, to do a lot of that stuff. Here's crystal green, canola plant, just 7 days old. You can see that as soon as it sensed the crystal green, it put out a pile of lateral roots and root hairs and melted. The crystal green actually drove right into it by putting out acids that can solubilize phosphorus out of the crystal green granules. For you guys that are mid-row banders, here's canola, and you can see where the mid-row band is, right here. Plants will tell you where it is, and they know where it is, and they pick that up by picking up the nitrate bloom from some of your nitrogen in the mid-row band, because nitrate is negatively charged, so it's mobile in the soil, and the plants really need nitrogen, so that's how they grow. I put this in here just to show you the kind of effect you can have from a granule. Now, MAP there's nothing wrong with MAP other than its salt index is fairly high, and you get a blast of ammonia that comes off the granule, monoammonium phosphate. So you get ammonium coming off, and it can be somewhat deleterious. So you take a granule of MAP and you put it near the germ end of a barley plant, and this is what happens. You put it on the other end of the plant seed, and this is what happens. So you guys that are single shooters, this is exactly what happens in the ground. To some degree, the slow up in the germination because of the salt index reduces water absorption. Remember what I said, if you got a high concentration of solutes in here, the water will move into the granule rather than move into the seed. So it absorbs the water from a surrounding seed and slows the imbibing of water, which leads to germination and also has a salt effect, somewhat of a toxic effect. So when you're single shooting, it's just sheer luck as to where that granule falls relative to the germ. And the same thing holds true for wheat. And the plant can do all this stuff. It can change the angle of the roots, the length of the roots, the number of lateral roots, the length of the root hairs, and canola is really good at that, and the density. And just think of it, the length you would think wouldn't be important, but when you're talking about phosphorus, potassium, and metal elements, just a fraction of a millimeter more length exposes or allows the plant to get more nutrients. And that's all run by genetics. Let me check my watch. Okay, we're going to have to finish here pretty quick. Just to show you what happens in the shoot, here's wheat. This is soybeans. Here we go, that vascular system runs into the leaf, and you can see the xylem and the phloem. This is a soybean leaf, and here's a wheat leaf. You can see it moves all the way from the bottom of the plant to the top, gets into the veins, gets into the leaf, and these are stomata on wheat on the underside of the leaves that open and close to allow that water to escape. And by the time it gets to the end, it's done. One tip for you, when you see the tips of leaves starting to turn brown and pulling nutrients out of here, tells you there's a shortage going on somewhere. And the plant is killing the cells and moving that into the veins. So the leaf tip and the edges are what are burning, or interveinal is where you're going to see the damage. Nutrients. Here are the key nutrients. Nitrogen and sulfur basically build the DNA, build the amino acids, and so on. These here are energy and cellular function: phosphorus, silicon, boron. These cations and zinc are what provide osmoregulation and signaling in the plant. And these, called the transitional metals, iron, manganese, copper, nickel, and molybdenum, they do the work. They're the catalyst that speed up the processes in the plant. And this is just a sort of a synopsis of that. But a plant is about 96% of these three: hydrogen, carbon, and oxygen, and only about 4% of all the rest of them. So that carbon and that hydrogen are really, really key in the plant. We all spend our time worrying about this. Well, we got to worry about that. And that's where photosynthesis and autotrophic plants have a role. In the soil, I don't know if farmers know this. In the soil, these nutrients here are negatively charged, so they don't stick to the soil particles, and they move, very mobile. These are partially mobile, and these are virtually immobile. And you notice them: phosphorus, copper, iron, manganese, zinc. Well, geez, these are all metals. Why would they be mobile? You can't dissolve them very easily. Inside the plant is different. Inside the plant, if the plant is short, it can remobilize when it kills cells. Remember, if a plant is turning, the tips are turning brown, it's killing the cells. It's killing the cells, they're dying, and the plant is solubilizing all the nutrients to move them. So these are mobile in the plant: nitrogen, phosphorus, potassium, manganese, molybdenum, and chloride. Immobile ones are boron, calcium, copper, and those metals again. I won't spend time here because I suspect Lyle has got this down in your area. Nutrient availability is largely depending on pH. You can see if you're around 7, you got yourself a really good soil. If you're about 6.5, that's maybe even better. As you move in both directions from there, you lose efficiency. And you can see here in Alberta, where we got a lot of acid soils, low pH soils, when you get down under 5, your efficiency in terms of uptake of nutrients really, really is depleted. And a lot of our soils need limestone to bring that pH up. Just to show you here and what we have for deficiencies, this was done in 2020. This was ANLF. 66% of our soils in the prairies showed low phosphorus, 78% low sulfur. That's why everybody's putting sulfur down. 37% low in boron, 55% low in zinc. And the tissues were 39% low in boron. I did a study where we took 100 wheat samples through 2020 seed labs, and we looked at germination and vigor, and then we did an analysis of nutrients in them all. Here's what happened: 100%, and this is wheat from all over Alberta, randomly taken. We don't know anything about the background of them at all. 100% were low in boron, 54% low in zinc. Just goes to show those maps are kind of right. And we have this going on: removal of nutrients. And I won't spend any time here because you guys can all access these same charts from our website in our portfolio booklet. But we have removal of all of these nutrients all the time, and I'll show you the micros. Look at the removals. We're taking grams. Every crop is taking that. We can see that zinc, for example, in wheat, this is the uptake and this is the removal. They take up a lot of zinc, and we're removing it every year. And we do that with canola, we do that with peas, we do that with wheat. all of these take up manganese. It's all leaving the prairies and leaving for somewhere else. Stress, the last thing. Here are stresses. I don't have to talk to you about stresses. Drought stress for you guys is a big one. Salinity is becoming bigger and bigger. Heat is another one. Well, when those plants are stressed, what happens? The stomata close and the oxygen is trapped in there because when the plant is photosynthesizing, breaking that water molecule, it's got to get rid of the oxygen. When it doesn't, it becomes dangerous and it starts destroying the fats, starts destroying the proteins and destroying the DNA in there and interrupts with this whole process of homeostasis or balance in the plant. So the plant is monitoring everything, biotic stress and abiotic stresses with all these kinds of little sensors in the plasma membrane of the plant. And it sends a signal. And one of the signals is that ROS. The ROS is useful when it's at low levels because it's a signal. If there's an attack on the plant signals that. It puts out a burst of calcium and signals that. So when you get that produced, you get hormones come into play to deal with things. The DNA comes into play. There's heat shock proteins here, and there's pathogen-related proteins here and defense genes. This all is going on, this communication crosstalk between hormones and sensors in every cell of the plant. And here's how it deals with the stresses. Antioxidants. Vitamin C, ascorbic is vitamin C. All plants produce vitamin C for this purpose. We use it ourselves as an antioxidant. Glutathione, a sulfur compound, we use it as well. Tocopherol, vitamin E, we use it as well. Carotenoids are those colorful things in plants. That's why we eat blueberries, strawberries, for those chloroplasts, or carotenoids. These are enzymes that are active. These are what are called compatible solutes when you want to have those solutes in the cell to retain water. The first thing the plant does, it makes proline, which is an amino acid, and shoves it in there. It gives it protection against freezing, in fact. Here are all the hormones that come into play, and we get down to homeostasis. Now, here's the key ones, and we're getting down to the nitty-gritty here. These are responsible for scavenging those damaged oxygens. Manganese sod, that just stands for that, iron sod, and a copper zinc one. Same ones used in humans because we're all eukaryotes. And the last thing I'll tell you is whatever you do, don't let your plants run out of K relative to stress. Regulates everything, moves everything, pushes osmotic pressures and so on. So here are the key ones: zinc, manganese, copper, iron, magnesium, boron, calcium, sulfur, and K for antioxidant enzymes and dealing with stress. Two last things. When a plant is going into reproduction, we get a change. Vegetatively, we move big time in terms of uptick in boron, zinc, manganese, copper, and calcium as we move in there. So we got to apply our nutrients according to the needs. And then the plant takes all of those things, all of those nutrients, shoves them into the seed so that the new generation has them in the embryo, in this case wheat, and in the endosperm, and in the seed coat. The only thing alive in a wheat seed is this little red line called an aileron and the germ. And look at what's in the embryo, the germ: sulfur, phosphorus, potassium, magnesium. Look at the zinc. Look at the manganese. Look at the iron. Look at the copper. And here's what happens when we germinate that. We get imbibing of water so that when we put on something like prime, the plant absorbs those nutrients as it's imbibing water. There's an initial start. It flattens out. We got to fix the mitochondria because the mitochondria are only alive in that aileron and in the germ, and it's damaged. Then it's got to synthesize more to provide that energy. We get that moving up here, and eventually we get germination where we get the radical form and the stores reserved. So this plant depends on the mitochondria for the energy to actually get going, and it multiplies as the plant grows. And that's the end. Oh, wait a minute. Wait a minute. Can you see that?
Yeah, I can see that just fine.
Okay. I threw these in here uh, uh, just to show you. Here's what prime is. This is PLS, specifically for peas and lentils. Look at what's in it. NPK, boron, cobalt, iron, manganese, molybdenum, nickel, zinc. No wonder we get responses to things like active prime. Here's prime, NPK, boron, iron, manganese, zinc. I started to build it at the beginning. Look at build, boron, manganese, molybdenum, zinc. I hope that, let's put it this way, I hope that after that long story, that your clients and our clients uh, understand why we have the nutrients in that package that we can apply during the summertime as foliars to seed as prime and PLS, specifically because legumes have to have different nutrients for nitrogen fixation. So that's all I have to say. And I'm hoping I didn't bore you to death, but I'm really passionate about utilizing these nutrients.
Yeah, I can tell you're definitely very passionate about it. Thank you very much. That was definitely some slides that I don't think I've seen since I was in college, and that was a while ago. So,
And many of the older farmers and still farming, you never had an opportunity to get even that exposure way back when. But when we were all in college, I took all that stuff 50 plus years ago, and it really didn't mean anything to me. It had no relevance to me. I just had to learn it to get through exams.
absolutely.
So we will be this will be posted on the website along with wheat, canola, and peas, and I'll get into the much more details. But I hope you folks maybe pass this on, because I think if you get this exposure and you understand and have confidence in the plant, it'll make your decision making hopefully a little bit easier, and why we get responses. But we need to have soil tests, and we have some tissue testing issues. And because if you don't know what the plant is short of, you may be putting on things you don't need. And if how do you make that decision? The plant will tell you. Even if you do just a few fields in your farm with tissues, you will learn so much by doing that. Anyhow, it's been right on the money, an hour is what I planned on. So watch for the additional stuff. And if you guys want to have another one of these, or we want to have a meeting down there and share this with a lot of other people, just let us know. Okay.
Sounds good. Thank you very much, Mike and Clayton, for another excellent presentation. A little science heavy on this one, but lots of our
Yes.
guys will probably really enjoy that. So I don't know if we'll have time for another one prior to the season, but
Probably not.
maybe me and Clayton will meet up out in the field and have a discussion and showcase some of the products during the spring.
You bet. Thanks a lot.
You betcha.
Thanks, Mike.
Okay, Clayton.
Thanks for watching, everybody. See you next time.